começar essa sessão, enorme prazer. E meu nome é Alfredo Gontijo e sou diretor do IEAT, está aí nosso banner, aí, Instituto de Estudos Avançados Transdisciplinares, e acredito que quem é da UFMG já internalizou isso no seu imaginário. Mas primeiro vamos compor a mesa e aí nós trocamos umas palavrinhas. Eu convido para subir à mesa a professora Maria das Graças Brandão, que é quem fez o convite ao nosso catedrático. E eu convido o professor, invite you to come here and take place. Com enorme prazer, então, é um enorme prazer que eu começo essa sessão e, inicialmente, como eu disse, falando um pouquinho sobre o IEAT, o professor Isao Kubo se encontra na UFMG, no IEAT, dentro do programa de cátedras FUNDEP e IEAT, e nós temos 3 versões: Ciências da Vida, no qual ele está inserido, Ciências Exatas, que em agosto próximo nós estaremos recebendo o professor Joseph Stuck, indicado pelo Departamento de Química, numa interface bastante grande, uma autoridade internacional na área de solos. Então, esse perfil, essa procura por interfaces disciplinares é a meta dentro desse programa. E dentro da área de humanidades, nós acabamos de receber a visita do professor Viveiros de Castro, que ficou conosco 3 semanas, era para ficar 4, mas na última semana, acho que o excesso de trabalho o detonou, e por recomendações médicas ele acabou tendo que ficar no Rio. Mas, na realidade, é que ele estava com uma cirurgia de olho marcada e o médico achou que era pertinente que ele não tivesse nenhuma atividade física ou de tensão maior. Então, isso foi acolhido, mas já existe a promessa dele de voltar para nos pagar essa última semana, e a presença dele foi extremamente rica, e, obviamente, que ele retornará outras vezes. Dentro do programa de cátedra, a visita múltipla é uma possibilidade. Por exemplo, o professor Bramer, neurocientista especializado em imagens do cérebro, em suas características funcionais, já esteve conosco agora, recentemente, questão de dias, pela 4ª vez, e já está previsto um novo retorno em novembro. Então, esses são os nossos catedráticos. Então, atualmente, nós estamos com as cátedras preenchidas, inclusive em Ciências da Vida, o professor Isao Kubo e o professor Bramer. Também para a ideia, os catedráticos, nós procuramos fazer com que a presença seja a mais rica possível em troca de interações, e na atuação dos nossos catedráticos, nossos convidados, como agentes ativos da promoção das interações e das aproximações diárias dentro da UFMG. Então, nesse sentido, nós procuramos fazer uma programação bastante intensa. E para o professor Isao Kubo, nós temos toda uma ocupação de 4 semanas, e vou dar destaque para alguns pequenos pontos, Mas a programação completa se encontra na homepage do IEAT, ou na página do IEAT. No dia 14, hoje, então, nós temos essa grande conferência, que é praxe, é a apresentação formal à comunidade da UFMG do professor catedrático. E no dia 14 nós vamos ter uma mesa redonda, que vai aparecer o cartazinho por aí, que é biodiversidade, conhecimento compartilhado e propriedade intelectual. Nesse caso, a expectativa do IEAT é sempre de trabalhar aproximações, convergências, unificações. Então, o que nós vamos procurar atender nessa mesa redonda, discutindo a questão da propriedade, são olhares diferentes, caminhos diferentes que, de alguma forma, devem encontrar convergências no futuro. Então, o professor Isao Kubo, como um especialista no trabalho de química, da bioquímica dos produtos naturais, trabalha isso numa perspectiva de obter esses produtos, de proteger, de comercializar. Então, a ideia da propriedade intelectual passa por trás dessa abordagem, que é a trajetória dele. Também participarão da mesa o senhor Eduardo Emerich, que é diretor da Biominas e que, portanto, também lá trabalha-se nessa perspectiva da proteção. E os outros dois são o professor Wagner Meira, do Departamento de Ciência da Computação, que é o nosso homem, o homem da UFMG, do software livre. Então, aí um pouco mais no contraponto, não do ponto de vista ideológico, mas do ponto de vista acadêmico, da abordagem do conhecimento. E estaremos trazendo o Dr. Fernando Matias, que é do Instituto Socioambiental, que então trabalha nessa perspectiva da interface dos produtos, das substâncias que são obtidas ali, na interface com a comunidade local. Então, essa aí, muito na perspectiva antropológica de como é que essa interface é feita. Então, acredito que deva ser uma mesa bastante rica. Além disso, ele vai trabalhar intensamente ministrando cursos ao nível de pós-graduação na Química, mas eu ainda daria destaque, todos os dias dele está ocupado muito na Faculdade de Farmácia, que é a referência disciplinar, mas eu daria destaque para um seminário geral na Faculdade de Ciências Econômicas, em que ele vai tratar dessa questão da propriedade intelectual, e o título é Patenteamento de Produtos Naturais, Situação Atual e Perspectivas. Depois ele vai para a Faculdade de Medicina e vai falar sobre o potencial da biodiversidade como fonte de novos medicamentos. E no ICB, potencial antimicrobiano de plantas medicinais. Irá visitar a Universidade de Viçosa e muitas outras atividades. Quem tiver interesse, e é óbvio que estimulamos a que todos façam essas interfaces, essas interações. Já aproveito também para fazer uma propaganda. No dia 22/6, nós teremos mais um encontro transdisciplinar, e dessa vez o professor Bruno Vanderlei, da Sociologia, um pouco na direção do professor Viveiro de Castro, e ele vai trabalhar a questão sociologia e em redes, e como usar técnicas de ciência da informação, tecnologia de informação, para reflexão sobre uma nova sociologia, o sociólogo do futuro, inserido nessa enorme complexidade do conhecimento hoje. Então, isso em termos de evento, em termos da informação básica sobre o professor Isao Kubo. A apresentação acadêmica é a prerrogativa de quem o convida, e a professora Maria das Graças vai fazer isso. Só um segundinho. Agora passo a informações mais burocráticas, e a maioria de vocês já deve saber, porque eu já falei isso em outros eventos, mas recentemente o IEAT foi institucionalizado pelo Conselho Universitário. Então, agora, o IEAT está instituído, fazemos parte da espinha dorsal da universidade. Somos um instituto de pesquisa que atua tanto nessa perspectiva de política acadêmica científica, quanto de desenvolver pesquisas. Tudo temporário, tudo temático, nada que se aproxime das nossas unidades acadêmicas com a sua rigidez. Como sempre, em decorrência disso, dessa institucionalização, nós estamos agora em processo de renovação do comitê diretor. Lá tudo é temporário, todos trabalham exatamente dentro do espírito de não se criar feudos, de não se criar estruturas que se enrijecem no tempo. Então, nós estamos no processo de renovação do comitê diretor. O comitê diretor atual já elaborou 5 listas tríplices que vão para o CEP, mas as congregações de unidade também têm essa prerrogativa. Então, fica essa informação que pode ser discutida. Acredito que o segundo semestre já começará com um novo diretor-presidente. O IEAT começou em 99 e eu sou o terceiro. Então, no segundo semestre já devemos estar com o quarto. Como sempre, a Eliane pede para avisar que quem precisar de certificado é só dar um nome para ela, que nós iremos emitir o certificado. Então, está formalmente aberta essa sessão. Enorme prazer tê-los aqui, agradecemos a presença de todos. E eu passo a palavra à professora Maria das Graças para fazer a apresentação do nosso convidado. Ao final, ele irá fazer a palestra e, ao final da apresentação, então, nós teremos a sessão de questões, discussões, como vocês julgarem melhor. É o momento oportuno para essas discussões e pode ser usado tanto a língua portuguesa quanto a língua inglesa. O Charles lá é um perito tradutor que consegue fazer isso de forma absolutamente brilhante, altamente elogiado pelas pessoas a quem ele presta esse serviço. Então, fica também o nosso agradecimento e o nosso registro. Então, professora Maria das Graças.
Então, bom dia para todo mundo, né? Então, eu só quero dizer rapidamente aqui, porque ninguém vai querer ficar me ouvindo, quer ouvir o que o professor tem para falar, é óbvio, né? Que eu estou muito satisfeita de estar aqui apresentando para vocês, meus colegas, para os alunos da universidade, o trabalho do professor Kubo, né, que é o convidado do IEAT. Eu quero dizer para vocês que eu tive o grande privilégio de fazer um estágio de pós-doutorado com ele em 98, lá na Universidade de Berkeley, e eu fiquei muito impressionado pelo trabalho do professor Kubo lá, porque ele é um cara, é uma pessoa das mais importantes hoje na área de produtos naturais, tem mais de 500 trabalhos publicados nessa área. E quando eu cheguei em Berkeley, né, uma grande universidade, era muito interessante ver que o laboratório dele é um laboratório pequeno, ele tem poucos alunos, então a gente tem essa mania aqui, né, no Brasil, de achar que grandes laboratórios, equipamentos, tudo é necessário para trabalhar a planta. Tá? A característica dele, que faz com que ele seja esse grande pesquisador com esse monte de trabalhos publicados, no meu entendimento, é porque ele usa e abusa das características de um grande pesquisador, que é, primeiro, criatividade. Né? Ele usa alta criatividade para poder desenvolver esses trabalhos desde o ponto de partida. Né? Observação minuciosa dos processos da natureza. Quando o professor Kubo vai pegar alguma coisa para estudar, ele já tem tudo arquitetado na cabeça dele, onde é que ele quer chegar. Nós aqui, geralmente, não fazemos isso, a gente tenta buscar sempre substâncias, né, ativas, publica esses trabalhos, como ele sempre me critica nisso aqui, que eu faço parte desse grupo, né, publica os trabalhos e depois fica tudo nas prateleiras, sem buscar essa meta aí. Tá? Outra coisa, né, que é importante falar, inclusive, principalmente para os alunos aqui, é a persistência. Né? Ele me revelou ontem que ele conseguiu publicar 20 papers em revistas de grande impacto com uma única substância que ele isolou. Né? Quer dizer, ele vai trabalhando e buscando cada vez mais saber mais sobre aquela molécula, sobre aquele composto, aprofundando, até ver que potencial realmente aquela substância tem. Né? Bom, e aí também, esses anos todos, né, esse tempo que eu passei com o professor Kubo lá e essa convivência com ele à distância, 2 anos atrás ele esteve aqui visitando a gente na UFMG, algumas pessoas o conheceram, né? Tá? Me fez admirá-lo também por outros aspectos como pessoa. Né? Primeiro, porque ele é entusiasmadíssimo com o Brasil, não só com a flora, como todo pesquisador é. Né? Todos querem estudar a flora brasileira e tudo mais. Não só com isso, né? Que realmente ele tem uma grande afinidade e estuda as plantas brasileiras. Mas também pelo povo brasileiro. Ele me revelou recentemente, agora nesse tempo, né, que ele admira muito o povo brasileiro, porque é uma coisa incrível. Só no passaporte ele tem 130 países visitados, quer dizer, ele tem visão de mundo total, de África, de Ásia, de América Latina, e ele fala que lugar nenhum do mundo tem pessoas igual o brasileiro, que pode estar passando dificuldade, mas ainda é generoso para ajudar o outro. Isso aí é uma coisa que só nós que temos, né? Então, ele consegue ter essa visão de nossa, né? Que eu acho que é um aspecto importante, tá? Então, ele me falou também, né, agora, né? Tem 30 anos que ele vem no Brasil, começou auxiliando os imigrantes japoneses na região de Belém, conhece muito a nossa flora, estuda a nossa flora. Então, isso é um aspecto importante, né, de trazê-lo aqui, que nós nem sabíamos, né, Alfredo? A princípio, era convidar um grande cientista para falar para a gente das plantas. Mas aí, antes de vir, o professor Kubo falou, olha, eu quero ter uma reunião com empresários, porque eu tenho produtos de plantas brasileiras que eu desenvolvi no meu laboratório, que eu quero que o Brasil produza. Então, isso para a gente é um grande lance, é uma coisa, é uma novidade, porque a gente fica aqui só arrancando os cabelos, que o estrangeiro vem, pega as coisas, leva embora, né? As empresas vêm. Ele não é um empresário, ele é um cientista, curiosíssimo com a nossa flora, e agora está oferecendo para a gente, né? Então, é um momento da gente refletir, né, em termos de financiamento de empresas, de investir na gente, porque, né, sendo aqui, né, imparcial ao máximo, nós temos que estudar essas plantas todas, tá? Então, esse tipo de trabalho, por exemplo, só para fechar, ele já faz isso na Nova Zelândia e no Japão. Tem produtos que ele desenvolveu com plantas da Ásia que são comercializados na Nova Zelândia e no Japão, tá? Então, a gente queria colocar isso, porque essa mesa redonda vai ser muito interessante. Eu acho que é um marco para o Brasil, para ter essa discussão, né? Tá? Que caminhos nós vamos ter, né? Porque a gente reclama demais, né? E aí, vamos ver o que a gente vai fazer aí, né? Trabalhar em cooperação, é o que eu tenho falado sempre com ele, que a gente tem que trabalhar junto, porque nós temos as plantas, eles têm a tecnologia. Para avançar mais ainda, é preciso trabalhar junto, não é isso? Buscar essa colaboração aí, tá? Então, eu fico satisfeita de vocês poderem, né, também compartilhar do conhecimento do professor Kubo e do que ele tem para falar para a gente. É isso.
Eu e Graça vamos nos assentar ali para poder ver melhor. Mas então agora eu convido o professor Isao Kubo para fazer uso da palavra.
Bom dia. Let me start explaining my connection with Brazil. That makes you easy to understand why I'm here. Okay? Of course, I'm still holding a Japanese passport by living in the United States since 1969, mainly in California. I had a very good friend when I was kids. We grown up together and we talk. And to the junior high school, we are talking about our future dreams. I was interested in becoming a lawyer, not scientist, and he was interested in becoming a scientist. So we dreams our futures. But unfortunately, he had a personal family problem because his parents, both parents, killed by car accident. Okay, and then he couldn't continue his education, so he went to immigrate to the Belém, Pará, Brazil, and we lost our contact almost 20 years. And one day, but because he still had, but he's not Brazilian. Phoned me in California, and he said, he called me Isao, please help me. Means he is now growing pimenta near the Belém. Okay, that was 1986 or 7, something like that. Because that time the pimenta grower near Berlin had a big problem with nematoda. Probably you remember that, okay? And he talked with our common classmate. Kubo is working in the entomology department at UC Berkeley. But you know, you have experienced the innocent people outside university always think university professor knows everything, right? That's not really true. So he said, help me. After 20 or 23 years later, then he invited me to come to the Berlin. Okay, so I started searching, what's the problem? I didn't know anything about the nematoda. But at that time, also the community of the not only Japanese, but the Brazilians, Japanese, who grow the pimenta, also officially asked the Japanese government to help this pimenta programs. So a group of the scientists, politicians came to the Benin. I was only one from the United States. So I was not officially invited. So actually, I couldn't do anything, and then I just had a nice time, nice conversation with him, and then this is the end of the story. Then, 2005, 4 years ago, until 4 years ago, I didn't have a contact with him again. Then he again phoned me and then asked me again, please help us. But this time, I don't know how to spell it, ABEP, A-B-P-E. That's the group of the pimenta grower, not scientists. Okay? And what's the problem this time? That I asked. And he said, we have a big problem with pimenta. That time he's kind of the leader of this ABEP group. Okay? And what's the problem is the exportation pimenta from the Brazil to the USA will have problems because when US customer select the pimenta as soon as arrived to the United States, they found the bacteria, Salmonella. So the US government warned to the Brazilian government, you have 5 years. You have to solve this problem. We are not allow you to export the pimenta to the United States. So they had the 5 years. Again, I didn't know anything about the Salmonella. Then we started but we do some microbiology. Okay, that's my connections. So many people say, you come to Brazil many times. But I come to Berlin, not really Brazil, Berlin, to see him. Okay? That's my connections. And when I started the chemistry, I'm a chemist working in biology department. Okay? So they're a bit different. The chemists in the chemistry department do the different type of chemistry. I do so-called interdisciplinary science. It's something like a borderline between chemistry and biology. Okay? And I try to give you that kind of example because when I started the chemistry, the chemists do not have extra time to do biology. Okay? Isolations, structure determination, synthesis. We have to put all our energy to play with chemistry. No time to play with biology. Somebody here may have that kind of experience. Okay, but young scientists may not. Okay, and then early 1960, Professor late Kapchans, probably if you're a chemist who do some natural, you remember him, his name. Okay, he started publications anti-tumor terpenoid, anti-tumor alkaloid. That's kind of the very unique the approach to chemists, because chemists didn't know anything about biology. Okay, so he isolated the chemicals. Well, we are working similar chemicals, but his compound looks like something special. anti-tumor or something. But if you think about the today, that's a very simple bioassay. Just I got the tumor cells, and then look for cytotoxic activity. Okay, but it's kind of a fresh and unique approach. So everybody start talking about the biological active natural product. Okay, that means the chemists they separate the natural product from plants or from microorganisms, and usually they send or ask the, well, our case, the NIH or something special program. So send the sample and then get the KB value or something, very simple cytotoxicity data. And then we follow the professor captions, or anti-tumor, such. Okay, that's the how interdisciplinary concept start. I'm talking about only my special natural product chemistry. Other part, I don't know, okay? Then, why I had a position in Japan, but I stay in the United States because, of course, I have the language barriers, but I like the freedoms. Can do anything, whatever I like. Okay, so you must have your own responsibilities. Okay, in Japan, never happens. The biology department hiring professor whose major is not biology, chemistry. In our department has the chemist, myself, and the mathematicians and physicists. Those people are doing their physics more close to biology. You know, not real physics for physics. Okay, so now really time comes to concern about the transdisciplinary field, because I'm trying to give you one or two examples what we can do with the interdisciplinary concept, and then some kind of limitations. Okay, so this is not really important, but I would like to start with this. Well, I know there are one or two here working in natural product, but what natural product scientists do today is, well, you don't need to read it anyway, okay? I just try to say do we have a pointer? We don't? Okay. Well, you can see the. I suggest you can use. Okay. You can see I just put the underlines, a red marker. The Yolanda and Dr. Ogura, they both are Mexican scientists. The Yolanda came from Mexico, from UNAM, Mexico City, and then she did work, but the plants were brought by the Yolanda. And she was working the anti-inflammatory compound from this plant. This plant called Anica, very common medicinal plant. We can buy even in California at the supermarket, because we have many Mexican people living in California. Okay, a usual case, like I said, they isolate the compounds from medicinal plants or microorganisms, do bioassays, and they make one paper. That you can count. There's so many papers being published. There's certain medicinal plant which they use in such place, and then we isolate the compound, shows such activity. Okay. That is a point I try to talk today, because so far probably Dr. Blondon mentioned her introduction about me. So far 14 natural products commercialized. Okay. I can say Chowdhury is the postdoc from India. Yolanda brought the large amount of this plant, and she was working in the anti-inflammatory compound. But I'm not adding here because she is still working on that the bioassays. Anyway, Yolanda need a compound to test her bioassays, anti-inflammatory activity. So I asked the Dr. Chowdhury, the postdoc from India, we need a compound, relatively large amount for further bioassays. As soon as he came from India, something like 4 or 5 days later, I asked him. And about 1 week, he spent his time in the library, and then he came to talk to me, and he said, I need other project. Because in the library, he searched about this plant, like using chemical abstract, something like that, and all chemicals being published already by German scientists. He is not very happy as a natural product chemist. He is expecting to isolate new compounds. That's the usual case. If you are biologists, keep your mind, okay? And I said, we are not doing about our generation need to go one step further. And then this is the biology lab. And I was not very sure, but I told him, I guarantee you can have many papers with this plant. Okay? So the information about Anico is available in book, written in Spanish or something like that. But it looks like a very important medicinal plant. So based on this information, we started the anti-inflammatory, many possible bioassays. And you can see we publish not really kind of the junk journals, okay? They are relatively good journals. Then, this is the 7 papers we published, and entitled the plant name Hazi. Okay, we have another maybe 10 papers, and not without the titles, the name of the plants. So something like close to 20 papers published with one plant, and then try to explain what the purpose or use by local people was correct. Now I'm trying to say something important, and then Mexican businessman started interesting because the one activity, something like the number one, the antibacterial activity, something like no one seems to be interested, but combines all biological activity. He starts thinking to make kind of cosmetic product or the kind of tablet. Okay, that's the way we usually do. That's how we send the commercial product with the medicinal plants. Okay, that's the kind of background, and then I started my main talks. When you work natural product, Brazil, when I was a student, I was told by my PhD professor, that was the 20th century, okay? He told us, to all students, the next century will be the era of the Brazil, because they have the most natural resources. That's still true. But every time I come to the Brazil, coming very slowly. To me, they're too slow. I'm sorry to say that. But well, examples. The Minas Gerais had the diamond before. I already mentioned to several people here. When you dig out the original diamond from underground, not beautiful. I don't know anybody have the diamond in a ring or something like that. And then the expert start cuttings and polish and then makes a beautiful the diamond. That has value. Okay? Natural product is similar, but the difference is diamond is not renewable. You just dig out and then sell it. I don't know how much it costs because I don't have any diamond. But the plants contains gene, means chemicals, natural products, and then need to cut and polish, means adding the biological activities. Okay? That's your responsibility. Scientists, something like the diamond cutter, or you need to make your compound from your natural resources important, interesting. I propose to discuss with the Brazilian businessman. That's the reason why he planned to have this round-table conversations. Okay. I come to Brazil, but I have only one paper with Brazilian plants, only one paper, because one of my friends, I just mentioned who lives in Belém, asked me to help analyze this plant. And that's the only one paper I have with Brazilian plant. The reason why I don't work Brazilian plant is I'm hired by UC Berkeley. I have a responsibility to report anything interesting to the university. Anything I use the university facilities, or even because I paid by university. It's all patent, well, not all, patent right goes to the university. So for example, I collect the plants or buy market and then do the analysis, find something interesting because I do the cutting, polish, and make the compound something interesting. That goes to the university patent. That is a kind of the problem you have. Okay, particularly company people were always protected by patent. I do have a lot of data. Probably I can do the disclose in the next round the table conversations. Okay, so that's enough to explain. When you work natural product, particularly from plants, I talk something on the good part because renewable natural source, you do have the most in the world. No question about that. But to work with natural product, particularly plant product, two problems: availability. Okay, they are like maintenance, good the anti-tumor compounds, but the plant grow in China. Okay, but the problem is they need this compound located not other part, but the mainly root. So to get the milligram sample, very costly. Okay, so availability is always you need to concern. And the activity is usually not very important. Of course, the certain activity is very important. Then the normal concept, the compound plant is someone can grow plants. That's one way. Many plants they grow. Okay. Or structure established. So sometimes synthesize the same natural product and then do some structure activity relationship studies. That's a normal way. And very rare case that we do the combinations. I try to introduce if I have time, because the medicinal plant contains the not one compound, many compounds. Sometimes this compound does not show the bioassay you do, but the compound synergize the compound showed activity, or sometimes antagonize. Remember, the Chinese medicine has the 4,000 years experience. That means the bioassay with the human body for 4,000 years. But no one can explain how did Chinese medicine work at molecular levels, because so many compounds work the synergize or antagonize. Okay, so we need to approach that kind of the solution, very simple ways. Then I do have several examples, so I try to show you the one or two examples. Okay, so first message, so far is the kind of a summary, and the first message is plant contains the many chemicals. So when talking about the biological activity, usually scientists do simple bioassays, something like a sweet or not, or something like this, and then isolate the compound which is responsible for the simple bioassays. Looks like scientists got answer, okay? But for particularly young scientists here, they try to think something from a different aspect. Then you can polish your natural resources. I'm not talking about the insect, but this is the examples, okay? The 1975, 4 and 75, I was international institute about the part of the United Nations, and then helping the African people with natural product. Means that's about time people start to really care, because that's the time the DDT or BH, those synthetic, the pesticide is out from market. Okay, so for politicians, easy to say you cannot use it because of the destroying environment. But they cannot say, then you have to use this. Even today, we don't have it. Okay, this is a kind of examples. This plants, I was given this plant. Actually, asked the director to work this plant to find the good pesticide, insect control agent, because this plant is free from insect attack in nature. I saw this, and then what we did is just tested the one insect and then got this diterpenes. Like the people still do, I reported we isolate diterpenes which inhibit the insect feeding, because it tastes very bitter, amargo. I don't like the bitter food. Insects do not either. Okay. But I moved to the United States and they start testing American pests. The same compound not active. And then instead the other compound, each time we do the different bioassays, I got the different compounds. So the conclusion is defense mechanisms of this plant against insect are multi-chemical. Okay, the many chemicals work together. Okay, so plant contains the many chemicals. That's keep your mind. And each compound was something the previous first paper, like the Mexican plants, the antibacterial activity, such the simple molecules. Okay, but same compound also showed various other biological activities. So multi-chemical and then multi-functions. I don't know today's biology can answer to those questions. Okay, each time I talk something like this conference and somebody asks me, give me your samples. We can test such, and then get the different result, or additional result. Okay, so plant defense mechanism against fungi, again, multi-chemical, and each compound has multi-functions. So we still have to do many things, but many people say the natural product is already finished. But in fact, we don't know anything. Okay, so this plan is really kind of the hope we can approach from different angles. Anyway, well, this is some addition, but it's not important. We published that papers in the chemistry journals, and then two groups. Whole structure should write, but anyway, because we publish in tetrahedron letters and the chemistry journals, we found a new diterpene which shows good insect feeding deterrent. Stop insect feeding because it's extremely bitter. And then groups, Dutch people, they synthesized this part. Well, this and the whole molecule is coming here. Okay, and the Japanese group synthesized this. Something wrong. Today, I can say that. Because they said that it have some activity, but not as strong as the plant extract itself. Because that's one of the 14 compounds which shows the activity against specific African Lepidoptera insect. Not all. And they tested, Dutch group tested the European pest, this is Dutch group. Japanese group tested the Japanese pest. Ecologically doesn't make any sense. Okay? So this is not important. Now, when I studied the natural product, not like today, we had only 60 megahertz NMR, so it's very difficult to establish structures, something like this. Okay, well, this is not important, but I'm just showing the complicated molecules. The compound isolated a few milligrams, and today we can establish those structures without not big problems, including absolute zero chemistries. Okay, there's something like this was published tetrahedrons, Journal Agricultural Food Chemistry, Journal Natural. It's published. Another one is the Bioessays, Journal Economical Entomology, something like that. Published in the first class the papers. Well, chemists should not be satisfied with those data because we do not have anything after DDT, or which many pesticide is out from market. We don't have anything. Many scientists work this. Okay? Get clear? Now, so what we do, so we need to approach different approach. This I have given the talk at the biology department here a couple of days ago. But anyway, these are very complicated saponins. When I was a student, almost impossible to establish the structure with 1 or 2 milligrams. Today can be done. Okay? These saponins isolated as the mosquito larvicide. They're very potent. They're more potent than the DDT. It's good. Now, remember I said that when work natural availability is a problem. Okay? This is the isolate from plant. The plants contains some amount of this, but how you can supply to the scientists, other scientists to do further assays? Impossible. Synthesis cannot be done. Okay? The combination means that you have to use potentiate the activity of this compound, but these compounds are not available. Okay? That's the usually end of the story. Isolate, establish structures with some biological activity. Chemists usually do this, or natural product scientists do this. Until my professor's generation, that's okay, because the main energy needed to put the chemistry, doing chemistry. Today, like I said, this compound, the structure determination, I'm saying it's very easy, but much easier than my professor's generations. Okay, so I do not have the real answer, but the one my propose is saponin is known as surface active compound or surfactant. The hydrophilic portions, hydrophobic portions, something like a head and tail structure. Plant extract, plant compound, almost all are similar. Okay, remember, not only Brazil, in almost any country if you go, they do have a medicinal plant. Medicinal plant never inject into your bloodstream, right? Because almost all plant products are surface active. So they destroy, if you inject your bloodstream, becoming toxic, destroy your red blood cells. That's the usual. Okay. However, these compounds have limitations, but simplest, the head and tail structure is another something like this. Very simple alcohol. Head and tail structures. Okay? And the tested against the same mosquitoes, which is important in the United States, not here. And then balance, head and tail length is a key because this is the surface active compound. So certainly when you're talking about the membrane of each organ is different. Okay? So activity is okay. Compared to the saponin, I just showed the previous one, that's very important, but this is 100 times less. But still, this compound has benefit because available, very cheap. And then those compounds, the alcohol is the most abundant organic compound in biosphere. Probably you know that. In particular in fruits. Okay? So relatively non-toxic to the animal. Also, most important to me, this compound very stable. One of the most stable compound. You spray in the Amazons because of the very strong sun irradiations. This compound is stable. Not the becoming logical or decomposed. Not like this. Right? So in United States now we have a program with Western Nile virus transmitted by the mosquitoes. Okay, so our university, because I did this work with the funding from California states, and they used their facilities. My facility is the university facility, so they got patent, international patent. So you cannot do business with this compound, but you can use without doing business, making money. But anyway, from saponin to alcohol, that's kind of the concept. It's one solution. I'm not saying this is the answer, okay? Maybe one possible answer. The available, stable, relatively non-toxic to the animals, and then cheap. Okay, the most of the problem we have with the natural product solved. Okay, now, you have this in your countries. Okay? And I don't have to explain, but if I giving this talk to the other country, like the United States, most of the audience do not know anything about this, the cashew apple. Okay? Everybody knows cashew nuts. In this country, in a certain season, if you go to like a buyers, you can enjoy these fruits, right? Okay, so the people who grow this one are, anyway, the nuts, they harvest, and then to make a commercial product, they need to remove the shells, cashew nut shell, very hard. So they heat it up and they make them soften, and then remove the shells. And if you visit the cashew nuts grower, usually small growers do themselves before they sell the product. Well, I'm talking about your countries. They visiting those growers. Usually, corner of the field, you can see the cashew nut shell is ready. They put the large amount, and then cashew nut shell oil is commercially available. But about 20 years ago, when I came to this country, my friend working in the Berlin took me to the Bahia because I do love the seafood, and he said Bahia is the best place. And then I saw this one, and then people do not know the value. And then some people squeeze out the oil and they sell cashew nut shell oil. Only cost $4 per gallon. very cheap. Okay? Now the scientists need to polish this the diamond. I'm helping you. Okay? So this from cashew apples, and the large amount of the cashew nut shell oil, which is the waste material in your countries, 2 compounds, cardol and anacardic acid. These are main compounds. It's a large amount. Okay? And remember, I said that when you people just heat it up and then making shells soften and then remove. And in that case, if you heat it high, this carbonic acid, the carboxylate becoming like cardanols. And so these compounds easy to isolate, even the tones material. Cashew is not original from this country, but original from India. Okay, so those oils, those compounds are easily available. Chemistry, those structure and the synthesis, including the many other the chemistry, were done by the Professor Late Taylor, the British chemist. He probably dedicated all his the life doing this the research of this type of compounds. So the chemistry will be done. Okay. I forgot the first slide. I said the plant contains the many the multi-chemical. I showed the African medicinal plant, and then showed the one diterpene oil. That's clethodim or argigarins. This is the anacardic acid. The first slide, I showed the 7 papers from made 1 papers. Okay, so we keep publishing biological activities of this type of compounds. And if you have a mark index in the library about yourself, most of the compounds we work are listed there. And you can see I didn't count, but our paper is listed as a reference. Okay, so hopefully my message: plant contain multi-chemicals and the multi-function. Just keep your corner of your heart somewhere. Okay. Anyway, this compound again, if you see, similar to the alcohol, head and tail structure. Head and tail structures. Okay. I don't think I have time to today to explain if this head is what the difference between alcohol, only OH, and this. Big difference. With many things are different. Okay. Maybe if the interest ask me, so I try to answer that. Okay. Anyway, there is availability is no problems in this country. Well, I don't know how much you need it, but available. And I didn't do anything in this compound. The UC Berkeley, so UC Berkeley does not get patent. Anyway, you can see I was talking about the alcohol. Well, anyway, it's not alcohol here. If you remember the activity, is it okay? This activity is about the 16 times more potent. Okay, and the cardols and without the tail part, only head part does not have activity, and anacardic acid the same. So it's more. So somewhere. Okay. Anyway, this tail part is necessary to have activities. Okay. I'm not talking today because I'm talking about natural product. Based on this, we are now synthesizing selecting the specific head and then tail portions. And if you look at my paper, our papers, recent papers, we're talking about the rational design of such. And because we're learning it from these natural products. Okay. Cashew nut crude oil, that's you can get from the nuts grower. still shows good activity. Okay? The availability is not problem. Here, in United States, we don't have it. We have to buy it. I don't know, the many Brazilians come to the Florida, maybe because of similar weather, okay? And there is okay? So there used to be, they use pesticide, including DDT. But the DDT is not allowed to use it. The mosquito came back. Not only that, the DDT is stable, right? So the number of alligator, that's the main things that tourists enjoy. Number of the alligator decrease because DDT has endocrine disruptor, means similar to the hormonal activity. So male alligator lost the, becoming like a female, lost the interest in the mating. So they decrease. So they really need to replace mosquito larvicide. And then the park ranger actually contact our university. But they are now trying to use alcohol because they don't have this one. I thought this is much better. But the material is not available in the United States, other countries, well, some other countries, yes. Okay? So this is something we can do. And remember I said that this, if head part is different, many things are different. One thing on this project, alcohols and then anacardic acid, the corridors. Activities against the mosquito is more potent. However, but I don't have time to explain this. The eukaryotic, prokaryotic cells are different, okay? And the fish is an important beneficial in water. So you need to kill the, but to me, if fish is there, why they use the mosquito larvicide? Because the fish eat the mosquito larvae. Right? But anyway, that's a requirement. We need to find the mosquito larvicide with natural product, but do not kill other beneficial creatures, including fish. So this has selectivities. This is natural product. Okay? Now, I'm changing subject because anacardic acid is available in this country. Anacardic acid is a kind of the diamond, just digging out from underground because it shows many interesting activities. As agrochemical, controlling the mosquito larvae, not involving big money, but I try to tell you the one important aspect which now some businessmen are interested. But in America, this material is not available. Okay, what's the interesting? When I have the slide, I can tell you. But based on this finding, we synthesize many series of corridor derivatives. We can put the different hydrophobic portions. Okay? This way we can. That's or the natural product would be nice food additives. Anacardic acid just published the one paper in the latest issue of the Journal of Agricultural Food Chemistry, coming from American Chemical Society. If you have that, we already published, so I'm not talking here, but that's good, very potent, the lipoxygenase inhibitor. Okay? Because anacardic acid, remember, anacardic acid is a good antioxidant without scavenging activity. Okay? If the scavenging activities, antioxidant becoming radical, so we have to be careful to use scavengers. Okay? Anacardic acid do not have the scavengers. That's not my main subject today. So I'm just saying the anacardic acid has various the biological activity. Today, we are now 3 postdocs now working the anacardic acid along the non-isoprenoid chains. Okay, anacardic acid and the cardol have a similar side chains, and then looks like a non-specifically inactivated many enzymes like a prostaglandin synthetase, xanthine oxidase, lipoxygenase, many. And looks like the hydrophobic portion, interaction with hydrophobic portion of the proteins, enzymes, proteins, and then this hydrophobic portion non-specifically bind and shows activity. Everybody, many scientists work the head portions. It's more elegant because that's, you can say, the active binding active site or something like this. But not to the hydrophobic portion. It's kind of the black, the spot. Now we try to synthesize the many compounds with different hydrophobic portions. We don't have an answer yet, but getting something interesting data. Okay, that's the reason why I keep continuous synthesize. Okay. I use the alcohol. Main purpose is to explain your product. Anacardic acid, cardol from the cashew is a good, would be good diamond, but they are easy to use the alcohol because that's the one that come from the saponin to the alcohols. Okay, so the alcohol is a non-specific surfactant, like a soap. Okay, depending on the hydrophobic portions, the alcohol has weak activity, but shows the toxicity of almost all organisms. Depending on potency, they depend on the side chain lengths. Okay, so the important things. This is just model. Here, this line is supposed to be plasma membrane, and this is the cell wall because this is the not plant but the yeast. Okay, that's close to the plant. But anyway, the cell biology usually draw like this one line the plasma membrane. Okay, and if you have a textbook of molecular biology, this is plasma membranes. That's not molecule, just line. Okay, so this plasma membrane is very important because plasma membrane contains various enzyme or receptor, and this is supposed to be membrane. It looks like better than just one single lines. Okay, transporters, receptor, something like that. So just keep in mind this one, and then use one of the specific the transporters, the proteins in membranes. This proton ATPase do selectively because they need to get energy. So they have a transporter to bring in glucose from outside into the inside the cells. And then start the glycolysis. When glycolysis start, produce a lot of the acid, you know this. And then inside of the cells becoming acidic. But usually inside of the cells neutral, because any proton comes out, they pump out. This is enzymes, membrane binding enzymes control the pH inside. Okay, now so this is a single line, but the proton ATPase is like this. It's been already established. Okay, I'm not talking too much, but the 147, this 148, this sustains in the membranes. It seems to be important because if the 148 sustain replaced by alanine, site-specific mutagenations, this enzyme lose enzyme activities. So something SH, free SH indicate important. Now, the alcohol, maybe I just skip this one because you have time. Now, usually membranes, plasma membrane, you say lipid bilayer, draw like this. If you have textbook, just see. Not more than this, okay? And then people call it molecular biology. I'm still alone, but every time I have a faculty meeting, all other professors are molecular biologists, particularly young one. And I said, you're talking about molecular biology, protein structure, something like that. That's not molecule, that's image. And no one helped me, but well, certainly this is not the molecule, but still understand for their studies. Okay, so I'm not against their opinions, but today's science cannot play with the molecules. But let me try to draw lipid bilayer, something like this. So little bit, not exactly the molecule, but close to the, more close to the real molecules. Okay, and how the enzyme is located, something like that. Okay, so this proteins, membrane binding protein, as you know well, this is not really turnovers or moving other place. Okay, it's sitting stably. So the hydrophobic interactions and intermolecular hydrogen bond interactions and electrolytic interaction. So the three type of the connections and the stables. Okay, now. This is the molecule big, but the alcohol is so small. What alcohol does this connections now? How about just picking up the one specific part of from here? This is the part of proton ATPase, and this is maybe the cholesterol, something like that. Okay. Now, alcohol is a weak activity. So why weak activity is alcohol approaching to this connections, because this is the intermolecular, but this neutral compound do not they come to the ionic portions. So this is not like a solid, maybe in water, oil becoming the grease. So this intermolecular hydrogen bond break by alcohols. This OH come here and then this break. That could happen. Okay. So this is kind of a summary. Alcohol disrupt the enzyme conformations by disrupting the intermolecular hydrogen bond. Okay, so anything like a surfactant have the similar activity. Different is if alcohol is approaching this something like this, is a small molecule. You cannot see the real big damage to the membrane. But remember, anacaridic acid, this head portion is aromatic ring. Okay, we can put the also the decalin moiety something become alcohol to prove this compound disrupt the membrane structures. Well, we can measure indirectly, but directly, if applied anacardic acid, this makes a hole because a big head. Okay, so you can measure the potassium cation inside from the cell start leaking. Okay, and if you have the naphthalene moiety larger, then make bigger, larger hole, like a punching hole, and then you can see the measure the protein or DNA, those cellular material leaking. Clear? Okay, that's the kind of the important things. Okay, time comes, so I'm just skipping the several things. I'm talking about the anacardic acid, right? So I do have this membrane interaction. I can explain more, but just probably enough. Then go to the anacardic acid, back to the anacardic acid. Well, before I go to anacardic acid, because anacardic acid is a natural product, this is the one of our projects synthesized, but these three are currently approved by FDA to use cosmetics or food additive as an oxidant. So this head portions and then tail portion different. Only certain balance, head and tail, log P not showing here. Certain balance then shows activities. Too short, too long, no, because that's the connection with membrane. Actually, I've already skipped. So long, the chain is important. Wow, this is not important. Okay, anacardic acid, this is the product from Kaju. In fact, today's waste materials. But I just mentioned about this is also good as a surfactant and to control mosquito. But more important, this is going to be good medicine too. Because remember, I just briefly mentioned these compounds inhibit the various enzymes. Not only that, because this structure is a surfactant and shows good antibacterial activities. Okay, and among the antibacterial activities, I'm just mentioned one very quickly. The compound shows good activity against the Staphylococcus aureus. Okay, Staphylococcus aureus is the bacterium which developed against many resistance against many antibiotics. And probably you have heard the methicillin resistance Staphylococcus aureus, MRSA. And then only one remaining antibiotics to control this bacterium is vancomycins. But some Staphylococcus aureus already developed vancomycin resistance. We don't have any medicine. Okay? This is the answer. Okay? This is the surfactant. The activity is, well, anything I skip, so I just briefly mention, okay? Try to understand. The resistance mechanism mainly two. One, this the present compound coming into the cells, inside the cells, so the bacteria develop the specific protein and then pump out. And sometimes this pump out the even unrelated antibiotics just outside, called multi-drug resistance. So that's the reason the Staphylococcus aureus survival against any antibiotics. Okay. This compound shows the activity against the all strains of the Staphylococcus aureus, but still activity is compared to the vancomycins, much weaker. Okay, so this could be good antibacterial agent, not like a human medicine, but the disinfectant. Antibiotics cannot use for disinfectant because we should limit to use the antibiotics because the bacteria is easy to develop the resistance mechanisms. Okay, I said the two main resistance. One is multi-drug resistance, and otherwise those toxic compounds coming into the cells, and then cell develop the specific enzyme, and then active compound inactivated. Mainly these two. Okay, anacardic acid is surfactant, so destroy the cells. How bacteria can develop the resistance mechanism against the surfactant? Not involve the protein. Okay, that's the reason this compound has a hope. That's the one, because it does not involve any enzyme to resist against this compound. That's the one good reason to use. Second, more importantly, I hope you understand. Just quickly read it if you have times. Well, hopefully the anacardic acid is one of the answers to solve these big problems now. Surfactants, remember that I explained with alcohol. That's the destroyed intermolecular hydrogen bond of the proteins, membrane binding proteins. Okay, multi-drug resistance enzyme proteins also membrane bindings. The anacardic acid works as surfactant, so something like. Okay, this is something similar to the proton ATPase. When you combine methicillin and anacardic acid, two, okay, test against MRSA, Staphylococcus aureus, which already developed the resistance mechanism against the methicillin. So methicillin alone does not work because methicillin comes into cells and then this multi-drug is pumped out methicillin to outside easily. However, anacardic acid works as surfactant. This is the interaction of plasma membrane and these proteins. So the methicillin is not active against the MRSA strains. But together with the anacardic acid modified their resistance mechanisms, so becoming active at original concentrations. Okay, other group also, American pharmaceutical company, other group also reported the same anacardic acid. They isolate from other plants, inhibit the beta-lactamase because the methicillin is a beta-lactams, one of the beta-lactams. Okay, so the anacardic acid destroys when methicillin approaching the resistance MRSA produce the beta-lactamase, which destroy the methicillin before they come into their cells. So maybe two beneficial effects of the anacardic acid. How they do this is interesting. Okay, that's the one thing. I hope we're not really getting tired. I'm trying my best, not wasting your time. Okay, so I'm back to the plan. When many scientists, including us, were reporting anti-tumor, antibacterial, antifungus compound, and just reported one anacardic acid. Again, multi-chemicals, multi-functions. For example, this is the surface of the olive. I'm sure there some Brazilian people eat the orange, and I'm sorry, olive. The olive tree, if you have an olive tree, we do have an olive tree in our campus. but American people do not eat much the olive, certain peoples. Okay, if you have it, they get the one leaves and then scratch the surface, and you can get the white powder. And then that's the part you see about whole entire leaves, but particularly back part, they're covered by the white powder. If you take the microscope photos, this is the normal microscopes. This is the electron microscope. And then today we can do this very easily. Get to the one leaves and then put in enamel tubes, and then take solid carbon 13 enamel, almost pure, almost 100% pure triterpenoid. So you don't need a column chromatography, just scratch, the easy chemistry. But does not show any antifungus activities. Okay, plant defense against fungi is not like anacardic acid. That's the one, the surfactant. Because this is you can see they covered all surface. So the fungus spore coming onto the leaves, they cannot germinate because this triterpene is not water soluble. So if you do follow the bioassays, isolating antifungus compound, you miss this one because it's large amount but cannot show activity. So plant defense against fungi, how you can transfer this knowledge to protect your crop. Think about that. Remember I said at the beginning, my product is commercial available. This is the one compound. It's available from the Brazilian plant, and the group of the Rio de Janeiro keep reporting the many papers about same compound as anti-inflammatories. Okay, anybody knows that probably. But anyway, this compound was isolated from the Japanese spice. You don't eat the raw fish like a sashimi, we call it. That's one of my favorite dish, Japanese dish. Very expensive here. Anyway, it's come with the fish, okay, spice. And this compound shows, this is a rather unusual chemical because it shows very potent anti-fungus activities. They're almost similar, but slightly less than amphotericin B. Okay, amphotericin B is the antibiotics, but highly toxic. If you buy amphotericin B for your bioassays, you have to put mask and then do everything, whole process in a safety box or something like that. That's the indicated. This is the spice isolate the spice people eat. Okay, but spice is so small, cannot isolate the enough availability is problem. Because easy to grow, but if grown in Japan, this becoming highly expensive because of small amount. Then we keep publishing these papers. Now, I don't know how he found, but the group of New Zealand scientists, they found these compounds from, I don't know, Science Finder or Chemical Abstract, or also Merck Index. This is the Merck Index as well. And then they found the large amount of the same compound from sawdust, the tree called Podocarpus, which is the important material, the tree to make this kind of material, furniture. Okay, so then when they cut, you have the dust, right? These compounds available large amount in New Zealand. Okay, this compound is good fungus compound, particularly against Candida. The problem, well, you don't have problems. The patients which are having the AIDS, the patients do not die because of AIDS, but the base they lose the immunities, defense mechanisms, right? So Candida is the one which causes the death, make death to the patients of the AIDS. If you find the website, polygodiol, not my name, polygodials, and then you can find the 4 or 5 the dealers selling the polygodiol capsules. That's coming from New Zealand. for AIDS patients, the Londons and Paris. You can find easily. I never found those products itself, but those people in New Zealand found the material. It's not available in Japan. Originally, we found it in Japan. But I don't know how good yield from Brazilian spice. Okay? So the availability is always the problem. That's the reason I'm showing example from Kaju. Because maybe other I can show, but from this country, that's probably the best so far. Okay, this skips. I remember to solve the problem with natural product, activities are weak. The last concept is maybe skip the combination, but I'll skip this the subject. If you have questions, I'll try to answer. I don't have the real slide, but last one. When I visited the Berlin is a place, not many things for tourists. Okay, we easily sit down somewhere and then drink the cerveza and talk. That's the only things we can do. And then my friend, they're always telling me what people use. And this is not the Brazilian plant, okay? But Dr. Blandon also worked the copaiba. That's a very common, if you go to the open market, the people sell the copaiba oils. And the copaiba oil also listed in market index, okay? And one of our late professor, who got the Nobel Prize, reported oil, copaiba oil is a good petroleum from plant. Because this is a compound, hydrocarbon, like a petroleums. But amounts, availability to run the car, I don't know how much you have. But the people in Berlin use, well, not everybody use this to in a soap, the good antibacterial activities. It doesn't show activity much, but the chemistry is interesting because all copaiba oils are very unusual. The sesquiterpene hydrocarbons, you can detect the more than 20 peaks and all molecular weight 204. We already established, and this is the major compound. Okay. Now, the copaiba oil shows good anti-acne agent activities. Got no activity against almost any microorganism, fungi, bacteria, but except the one, P. acnes, Propion bacterium acnes, that's the bacteriums. Remember at the beginning I said the Brazilian scientists needed to polish the natural product. Okay? So those compounds shows only antibacterial activity against only P. acnes. Okay? So, but those people in Berlin do not know, but that's what we found. Then to develop, because this your material is not available large amount in other countries. We do have in States the copaifera, but there's very small the oils in the Berlin areas. The big copaiferas and bore the holes, and then put the cerveza bottle, something like this, and then oil start coming out. That's how people collect it. Okay, now how you polish? One answer to cause the acne is 3 reasons. One, because of this bacteria. So your acne is caused by the bacteria. Copaiba oil may cure. Okay. Second reason, who has acne? Probably you do not have it. Young boy usually have the acne because this is caused by the testosterone, male hormones. So you need to inhibit the testosterone synthesis. Tetracycline does too. The tetracycline is known antibacterial agent and then inhibit the testosterone synthesis. How about natural problem? So if you really have a problem with acne, they go to see the doctor because so much the problems. Maybe copaiba oil is not strong. I don't know. Okay, so the copaiba oil does not inhibit the testosterone biosynthesis. So you need to combinations. So that's I call the polish. They make good cosmetic product. I don't have time, but you have many things. Angiroba, if you go to the market, it's interesting to see. Okay, and then well, probably you do have the similar medicinal plant in this state, well, whole entire Brazil, because Brazil has the most renewable natural resources in the world. And scientists need the polish to make a good diamond. Okay. Lastly, I have to say obrigado. I hope I am not making your waste, your time is waste. Hopefully, you can get some kind of idea for your future studies. And I will be available until 21st. Probably he already mentioned. If you're shy to ask a question in front of people, just want to talk to me, I'm available. Okay. And any things you want to discuss with me or comment, more than always welcome. Thank you. Obrigado.
Muito bem, gente. Então, nós agradecemos ao professor Isao Kubo. Nós agradecemos pela brilhante exposição com relação aos produtos naturais, isolados, e nós abrimos a sessão para perguntas. E aí, sintam-se à vontade para formulá-las em português, inglês, escrita, falada, à vontade. Então, está aberta a sessão. E tem um telefone, um microfone sem fio aqui, que pode atuar como telefone.
Bom dia. Eu sou o Wagner, professor do Departamento de Clínica, Patologia e Cirurgia da Faculdade de Odonto. Trabalho com microbiologia e patologia e a gente desenvolve alguns trabalhos em fitoterapia com a professora Maria das Graças. E eu gostaria de saber do professor Kubo se ele faz algum trabalho com micro-organismos da cavidade bucal, se tem alguma planta de interesse aqui no Brasil que ele gostaria de trabalhar e quais as perspectivas que tem nesse trabalho com micro-organismos da boca.
I wish I could answer in Portuguese, but let me explain in English. I didn't mention anything about the Streptococcus mutans or gingivalis, something like the microorganisms important to cure the mouse. Well, anacardic acid shows very good activity against S mutants. and weak activity, gingivalis. Okay, and almost one American company tried to use anacaridic acid as a toothpaste. But again, the problem was the availability. So it's not commercialized yet. But most of the compound I just mentioned, the antibacterial activities, it shows activity against the S mutants. The three main the microorganisms important for you probably: gingivalis, the S mutants, and the actinomyces. Anacaridic acid is kind of perfect.
Ok, mais por favor.
My question will be in English. I'm Dr. David Bird from Canada, just visiting in botany. This is a more interdisciplinary kind of question. I wonder about the danger of, or do you have any response to the potential problems of so-called bioprospecting, where people from more developed countries come and it's them who develop the natural resources of countries that don't have the resources to do it? Do you have anything to say about that?
Actually, I'm not involved like that. That's the reason I'm not collecting plant myself. I'm not botanist. I don't know much about the plant. But I was about 10 years a consultant for World Bank. My territory was both Thailand and Indonesia. Instead of giving them money directly, they try to find out same thing like I mentioned here as about the Brazilian natural resources. Indonesia is the second largest rainforest, having second largest, following the Brazil's. And there are many, many interesting the plants. But I don't know which one is endangered. So it was very difficult to find which plant is interesting. But of course we collaborate with the local scientists. Particularly Indonesia was the plant taxonomy was well done because Indonesia was once the Dutch colony, and Dutch people did a very excellent classification of plants. Well, many Indonesian people use, related to the first questions, the neem stick. Neem is the original from India, and today's many scientists believe that's a good pesticide, insect control agent. But in India, it's a kind of medicinal plant. Not only India, but it's available in Indonesia, not Thailand. They chew very strong bitterness. That's how well, today young people start using the Colgate or whatever. It's made by big industries. But Indonesia and Thailand, maybe I'm not answering to your question directly, but Thailand is affected by Chinese culture. Indonesia is an Islam country, very close, geologically very close, but as far as the medicinal plant are concerned, totally different. The same plants, but different name, use a different purpose. I cannot explain how they use this. But, I don't know how many species we keep losing, once because I keep saying that I'm going to Berlin areas, and every time, well, not in Berlin city, but outside of the Berlins, every time they have a new babies, they cut down trees in the forest, certain space, and then cultivate the mandioca. That's enough to eat one newborn babies. They start as soon as they have babies. I know the discussions, the many the things happening, but people, the American people say in United States and Brazilian people in varying areas, and then people, but I have a friend who is also working in USPE, in São Paulo areas. People say totally different things, okay? But as soon as I came here, the London took me to outside. It's a very nice place. And she said the eucalyptus. But I understand that until she explained to me, I didn't know the forest here is secondary, not the original. And someone who grow the eucalyptus, because that's fast growing trees, and then sell to make paper, pulp, she said, to Japan. She tried to complain to me. But someone do this to make money. I cannot say to this Brazilian, don't do that. You know, it's difficult. But so this kind of economical balance, that's the reason I'm not really touching the real plants, which I don't know. The copaiba oil, I don't know who collected, but if you go to Berlin, you know, many the local market sell this. And Copaifera is an endangered species. As a chemist, very hard to, especially my language barriers. Probably I didn't answer to you very straight, but that's the only things I can say now.
Yeah, thank you very much. I am Professor Ruben from Chemistry Department. My question is about the intellectual property and technology transfer issue that maybe are you going to talk on another next talk in the future. But I would like to know, because you mentioned that you work with this kind of interesting kind of compounds, and I would like to know exactly from your lab or from your experience or from your point of view, how many compounds do you have that are patent and was transferred to the industry, if you have? I would like to, you mention a little about that from your point of view.
I have no patent about compound. No. I do have a patent, but biological activity or something, not myself, but I have my responsibility to report to the university, because I do the research in my lab. Because of the electricity, those basic things, and the university pay for that. So not my patent, but well, probably you know the United States, the law has been changed. And used to be professor get no money from patent, but what's the law, about 10 years ago, or 20 years ago, Brown law or something, I don't remember. But anyway, even my research is supported by NIH. Used to be patent goes to the NIH, well, government. And that was the real inactive activity with patent. Now this patent, not 100%, in part, come to the university. The University of California now have 10 campuses. Okay, so they were a little bit tricky. When we report, they have a bulletin board, you know, something like for industry. You know, now sending by email. We found such, and then wait 1 year, open mainly the company located in California. And if no company shows interest, open up the entire nations. Okay? After 3 years, no American company showed interest. They open to the international. And then if company showed interested, university asked to pay patent fee or everything. That's, I said, very tricky. So I don't have my own patent.
Mais alguma pergunta? Eu gostaria de fazer uma... Eu vou tentar formular uma pergunta transdisciplinar. Primeiro, no eixo da complexidade. O senhor mencionou o diamante como algo simples, as estruturas que o senhor isola como algo de relativa complexidade, por exemplo, as saponinas, e, finalmente, eu gostaria e posso pensar no homem como o topo da complexidade, no fenômeno da consciência. Numa outra direção, numa outra reflexão, eu posso trazer a ideia de isolar os produtos, que é a sua opção, que é a opção do senhor, e a ideia da análise, e aí no mesmo contexto. E talvez no contraponto, a ideia da síntese, sintetizar esses produtos em laboratório. A terceira vertente seria a vertente dos mecanismos que são utilizados, sejam pela natureza nas plantas para sintetizar essas substâncias, seja no laboratório em termos de tecnologias de síntese. Se eu penso nas estruturas simples, como é o caso do diamante, então nós já temos todos esses ingredientes dominados de uma forma bastante simples. Nós temos o diamante feito na natureza e temos o diamante feito no laboratório, o mesmo diamante. Então, aqui a convergência, a unificação é bastante plena. Se eu penso no outro extremo, o homem, aí a unificação fica extremamente complicada e difícil. Então, eu entendo que o senhor está trabalhando numa posição, numa opção de gerar conhecimento que é extremamente privilegiada, porque ela tem um grau de complexidade, que é o grau da complexidade ainda manipulável, tem, no caso do isolamento, da análise, tem a perspectiva de fazer a síntese e tem a perspectiva de estudar os mecanismos com que esses produtos naturais são sintetizados na natureza ou no laboratório, através de tecnologia. Então, nesse complexo cenário de parâmetros, como é que o senhor se insere, como é que está o diálogo entre quem está analisando e quem está sintetizando, aonde está o estado da arte, do grau da complexidade, da síntese no laboratório. Eu perguntaria objetivamente, é possível sintetizar no laboratório produtos como as saponinas? Uma pergunta transdisciplinar.
It's a difficult question to answer. Anyway, the people, not the people here, but the innocent people today, really looking for natural product. Okay, so they're talking about the synthesis, but let's see, the natural product has a broad market. The medicine is, for chemists, probably easiest part, because even we don't have to worry about how much it costs to synthesize, because that's the balance of somebody's life or cost. So people pay even $1 million, I don't know. But anyway, cost is not important, and the toxicity is still somehow acceptable. The balance. So synthesis is still main part play human medicines. Okay, but the natural product are really hoped to play very important role in cosmetics and food industries and agrochemicals, because the medicine is for sick people, so there's some risk, just balance. But the food, well, once people establish their life and then having nice life they certainly want, but women want to be beautiful, cosmetics, and live longer, and health products. And now, I don't know the Brazil, but if you go to big supermarket, always in the States, you have the corner which sell the vegetables and fruits, say organic, more expensive. But the people, even more expensive people buy. Why? Because they care about their health. So people really waiting for natural product, but I talked very pessimistically. You know, the many pesticide is already out from market, but we don't have anything. Okay? And company are not interested. United States, the natural product used least in the world, because the people did not care until, I don't know, recently. Because all the agriculture is big, so most of the pesticide was used. But now the Dow Chemicals, Monsanto's, those chemical company used to make tons of pesticides. It's now really bad shape, because the people are not accepting the pesticide. But not only the United States, my friend works for Sumitomo Chemical Company, and used to be, he was very good because good salary, but he's really worried about the market. Now, synthesis is still plays I'm repeating, the main part of the human medicines, but not anymore the cosmetics of particular food, agricultures. That's the reason I said that this country has the most natural resources, just need to polish. And laboratory synthesis, natural synthesis, if the same compound, you cannot say completely identical. The many compounds, but that can be improved because the many compound synthesized, after like us, the reported compound shows good activities, not available, and they synthesize. But they always contaminate the other compound. Often those contaminant does the side effect, something like this. So but yourself, think about you have something like this water, let's say yogurt, and see colors, natural or synthetic something, which you choose. That's our society react now.
Mais alguma pergunta? Então, nós agradecemos de uma forma bastante enfática a visita que o professor Kubo faz ao Brasil e ao UFMG, e esperamos que essa seja somente a primeira de uma série de visitas e que essa posição, nessa faixa intermediária da complexidade, faça com que ele seja um instrumento de promover interações entre as áreas do conhecimento aqui dentro da UFMG. Muito obrigado, professor Kubo, muito obrigado a vocês.